All right. Hi, everyone. Thanks for being here. I'm Aditi Sathe from the Vanderbilt University Medical Center, and I'm really happy to have the opportunity to present here. I have nothing to disclose. Starting off with a quick primer on Alzheimer's disease, it is a progressive neurodegenerative disorder marked by memory loss, cognitive decline, and behavioral changes. It is the most common cause of dementia in older adults and is characterized by two primary pathologies: amyloid plaques and tau tangles in the brain. Amyloid beta peptides accumulate as oligomers in the extracellular space, and these oligomers assemble into amyloid beta plaques over time. This interferes with neural function and triggers immune responses, such as microglial activation and neuroinflammation. Inside the neurons, hyperphosphorylated tau proteins form neurofibrillary tangles, which ultimately leads to cell death and tissue loss. Their neurodegeneration manifests itself as a thinning of the cortex, shrinkage of the entorhinal cortex and the hippocampus, and a notable loss in brain volume, eventually resulting in progressive cognitive decline. AD is a progressive disease in that it starts in the entorhinal cortex and hippocampus and then slowly spreads to other parts of the brain. Here we see a simplified continuum of AD involving various stages that unfold over an extended period. During the prolonged preclinical phase, which can last for up to 20 years, individuals may appear symptom-free but are undergoing significant neurological changes, including the accumulation of amyloid plaques and tau tangles. This stage is crucial for understanding the disease's onset and progression, providing a window of potential early interventions aimed at slowing or halting the disease's trajectory. AD has traditionally been thought to be a disease of the gray matter, but research has consistently shown that there is white matter atrophy independent of gray matter, and that white matter changes are detectable up to 22 years prior to disease onset. Potential mechanisms for decline in white matter integrity include axonal loss, demyelination, microglial activation, and inflammation. Prior AD studies have found that white matter changes in specific tracts are associated with diagnosis along the AD continuum, higher levels of brain amyloidosis, and exacerbated tau accumulation. White matter tract atlases provide standardized references for identifying and analyzing specific white matter tracts, facilitating comparisons across studies and populations. The tract templates used in the current study are freely available online from our group. This includes 48 tracts across 7 anatomical groups, including the association, limbic, projection, occipital transcallosal, parietal, motor, and prefrontal transcallosal tracts. Neuroimaging techniques like diffusion tensor imaging allow us to study white matter microstructure and potentially detect alterations early in the AD continuum, even as the person remains cognitively normal. Here on the top panel, we see a schematic representation of standard DTI-derived metrics. Axial diffusivity describes the diffusion along the principal vector, radial diffusivity perpendicular to the principal vector, and mean diffusivity represents the overall magnitude of water diffusion independent of anisotropy. Fractional anisotropy is a scalar value between 0 and 1 that describes the degree of anisotropy or directionality of the diffusion. On the bottom panel, we see a typical FA map with higher values in white matter and a value closer to 0 in the cerebrospinal fluid, where diffusion is largely isotropic. Partial volume effects are a common source of error in DTI imaging and tractography. At the bottom, we see an example of free water partial volume effects at the interface between brain tissue and ventricle containing cerebrospinal fluid. The image on the right depicts an ideal slice in which the brain tissue and CSF are clearly delineated, while the center image depicts the contamination in voxels that contain both CSF and tissue. By incorporating free water correction in the conventional diffusion modeling pipeline, we can obtain tissue-specific indices for voxels contaminated by free water or any other tissue heterogeneity. This is accomplished using a bitensor model that has 2 compartments: the extracellular or free water compartment, characterized by an isotropic tensor with the diffusivity of free water, and an intracellular compartment modeled by a diffusion tensor. Each of the DTI metrics, including free water, have specific biological correlates that are largely driven by inflammation and degeneration in the brain. Amyloid beta plaques trigger microglia to initiate inflammation, and the inflammatory cytokines in turn damage tissue, contributing to neurodegeneration and increasing free water. The degeneration involves demyelination and loss of axonal integrity, leading to decreased anisotropy. While previous research has shown that global white matter alterations are associated with different clinical and biomarker categories of Alzheimer's disease, their topography and specific relationships with cognition remain unclear. With this study, we had two primary research objectives. First, exploring white matter microstructure links with cognition in a large longitudinal cohort. And second, identifying specific white matter tracts that are the most sensitive to cognitive decline in aging in AD. We used a linear mixed effects regression model with covariates including age, age squared, education, sex, race, ethnicity, diagnosis at baseline, and ApoE2 and ApoE4 status. We also controlled for age converter, age square converter, and age microstructure interactions. We leveraged a large longitudinal cohort of about 9,500 sessions with over 4,500 unique participants across 7 different studies. The average age at baseline was 74, with about a third of the participants converting to AD over the course of the data collection. To reach this sample size, we have been working as part of the ADSP Phenotype Harmonization Consortium to harmonize rich endophenotypic data from over 40 different studies. Here we have an example of how the memory scores are co-calibrated. Anchor items are the tests that have identical content and administration methods across studies, and the additional tests are used to derive item parameters and estimate factor scores for each study. To harmonize across different MRI scanners and cohorts, we used a technique called longitudinal combat that implements an empirical Bayes method in a linear mixed effects model framework. Moving on to the results, we found that free water is significantly associated with decline in memory performance across all 48 white matter tracts, with the middle frontal gyrus transcolossal tract presenting the lowest p-values. Additionally, 8 of the 10 tracts that were the most sensitive were transcolossal tracts, highlighting the importance of these interhemispheric connections in age-related cognitive decline and AD. Here's a heat map showing the associations of the free water corrected DTI metrics, including axial, radial, and mean diffusivity across all 48 tracts. You can see that free water in the top row, particularly in the limbic, prefrontal, and association tracts, exhibits the highest correlations with memory performance. We also found that free water is significantly associated with decline in executive function performance across all 48 tracts, with once again the middle frontal gyrus transcolossal tract presenting the lowest p-value. Here as well, 8 of the 10 tracts that were the most sensitive to decline in executive function performance were transcolossal tracts. And here is the heat map showing the associations of the free water corrected metrics with each tract for executive function performance. You can see that free water in the prefrontal tracts exhibits a very high correlation, and that the pattern of associations is more spread out across the tract groups as compared to memory performance. To wrap things up, we found robust associations between cognition and white matter microstructure metrics, particularly with free water corrected measures. Free water in the middle frontal gyrus transcolossal tract, IFOF, and caudate to middle frontal transcolossal tract seems the most predictive for decline in cognitive performance in the aging population. Ongoing work is exploring machine learning approaches to examine feature importance for tract-specific DTI metrics. Here are the preliminary results showing the 10 most important free water corrected measures in a linear mixed effects model as ranked by XGB tree. We are also looking at combining free water corrected measures from specific gray and white matter regions to develop and validate an advanced free water imaging signature. And finally, we are working on incorporating additional harmonized data in the form of new cohorts and different composite cognitive scores for domains like language and visuospatial functioning. With that, I'd like to thank all members of my group, specifically Dr. Derek Archer, who couldn't be here today, and all of our wonderful collaborators for supporting these large-scale harmonization efforts, and thank you for your attention.
Thank you very much. That's a very clear, very easy to follow talk. We have one question on the app, and certainly if others have questions, feel free to put them there as well. We have about 2 minutes to go from the questions. So the first one's quite simple. Why would you add age squared into your statistical model?
Yeah, so we added age squared to take care of the nonlinear age effects in the model. Generally, the nonlinearity as it relates to white matter microstructure is more important or at least more conspicuous in ages under 50, but we did want to be robust to those kind of interactions and changes as well, so we added the H-square term.
So maybe subsequent to that, you have a lot of interactions in that model which you don't decompose at all, which could really be interesting and tell us about kind of very specific trajectories of decline based on age or sex. Do you have any thoughts on what they might have shown?
The current analysis, we didn't do any interaction analysis for this specific study, but that's definitely something we're looking into.
Like the ones that you already showed that were in your model, you haven't looked at kind of where...
Yeah. So we did have the interaction terms in there, but we didn't get to that specificity that we're looking for with specific tracts if they are interacting with age, yes or no, and those p-values. We were more interested in the cognitive scores at this point, but in the future, that's definitely something we'll be looking into.
Okay, great. Thank you very much. Join me in thanking the speaker for excellent presentation.